Balchem has spent decades perfecting the art of microencapsulation for both the human and animal nutrition segments. Add in food processing and nutrition expertise, and we have the complete package you need to overcome the formulation or production challenges that hinder innovation. Look to PetSure from Balchem to provide the expertise and capabilities that redefine entire product categories and help bring your imagined products to reality.
Hello, everyone. My name is Scott Sorrell, Director of Global Marketing for Balchem, and welcome to the Real Science Lecture Series. Today, we're taking an in-depth look at pet food diet formulations and how to manage some of the more unique compounds to ensure we're delivering the best nutrition. Dr. Kate Shovler from the University of Guelph is here today along with Cindy Banton to walk us through their latest research. I would now like to introduce Dr. Kate Shovler from the University of Guelph. Dr. Shovler is an associate professor in the Department of Animal Biosciences. She received her undergraduate degree from the University of Guelph before joining the research team of Drs. Ron Ball and Paul Penchars at the University of Alberta for her PhD. Following her PhD, Dr. Shovler spent 8 months as the provincial equine specialist for Alberta, and then returned to Guelph for her postdoctoral fellowship in companion animal nutrition. Kate spent 8 years in the USA working for Procter & Gamble and then Mars before joining the faculty at the University of Guelph in 2015. Her current research focuses on energy and amino acid metabolism in monogastric animals, primarily cats, dogs, and pigs, providing information for feed manufacturers and for others who work with animals to improve the health and welfare of pets and livestock. Kate also works with sled dogs and service dogs to improve their health and nutrition as well. Dr. Shovler is joined today by Cindy Banton. Cindy recently completed her master's degree with Dr. Shovler in companion animal nutrition. Her research focused on grain-free diets in dogs, including supplementing a grain-free diet with micronutrients and investigating consumer habits toward grain-free diets via an international survey. Sydney is currently working with the Shovler Laboratory as a graduate research assistant. Kate, you should now have control of the screen.
Great, thank you very much. I just want to do a check and make sure that everybody can hear me clearly. Just a thumbs up. Great.
Yes, everything's fine.
Thank you. All right. It is absolutely my pleasure to be here. Thank you very much for the invitation and also the opportunity to talk to you about what's going on in my laboratory. And this is just one snapshot. And to give Sydney the opportunity to share her absolutely outstanding work that she completed during her master's. So today we're going to focus on amino acid or protein quality, and then I'm going to start there, and then we're going to move into the studies that Sydney did during her master's, namely one in vivo dog study, and then a global survey that she'll go and talk you through on the grain-free consumer. Just to start, I always like just to share any conflicts of interest that I might have. I currently am a member of the Trial Companion Animal Scientific Board, and my research is funded by Champion Pet Foods, RC Hagen, Smart Earth Camelina Corporation, FL Emirate, the JM Smucker Company, Saskatchewan Pulse Growers, the National Science and Engineering Research Council of Canada, the Canada First in Research Excellence, MITAX, and Agriculture and Agri-Food Canada. Pictured here are the postdoctoral fellows, research associates, and graduate students that have all contributed in part to things that you're seeing as we go throughout. On the far left-hand side is myself and my former postdoc, Wilfredo Mansilla, who was integral in our protein quality and our DCM work. Lisa Fortner, who is a research associate at Mars, Kara Cargo-Froome, who is a PhD student with me, Dr. James Templeman, who did his PhD and is now a postdoctoral fellow with me, Dr. Julia Pozzali, who's doing her PhD with me and is originally from Brazil and completed her master's with Greg Aldrich at Kansas State. And Michelina Crosby, who is a PhD student with me and had previously done swine research in her master's as a prelude to coming into my lab using similar techniques. And last but not least is Sydney. And Sydney recently completed her master's with me and is working this summer to help support further research. So a little bit of a background for today's talk. I'm going to describe and talk through protein quality with you, talk about some amino acid requirements. Specifically, I'm going to talk about methionine requirements, but I'm also going to talk about secondary metabolites of methionine. And that's what's really going to bring us into Sydney's work, where she's going to talk about what DSAM has taught us and where we're going after her considerations of micronutrients and sulfur amino acid supplementation, and then the potential consumer's role in the etiology of this disease. So to get us started, protein quality is well defined across all mammalian species, and it includes not just protein content, but the amino acid composition of that content and the digestibility and the bioavailability of that diet. Now, of course, points 1, 2, and 3 are all attributes of the food. And while we need to quantify those on the food's basis or on an ingredient basis, which is what we do in human nutrition, what we do have to do is we have to compare them to the requirements of the species of interest. And indeed, for your own knowledge, if you go to the grocery store and you're looking for different complete products that would be on the shelf in the middle and not on the circumference of your grocery store, these products may carry protein quality claims, such as a source of, a good source of, or an excellent source of protein. And when those are when that is defined, those are defined on a regulatory basis. And what it is the protein content, the amino acid composition, and the digestibility together in an algorithm that is compared to the amino acid requirements of 4-year-old children. And that is used to define to the consumer the quality of the protein. And so I'm going to reapply these concepts as we go through and we examine what data we have in pets to help us look at that. So first and foremost, let's look at protein content and amino acid composition. It's always important to first start with the protein composition and amino acid content of our commonly used ingredients. What's important to know is we have ingredients such as rice that we commonly call carbohydrate ingredients. However, all ingredients carry all of the macro and micronutrients to a certain extent, and our job is to really combine them as formulation scientists or product developers so we can achieve the amount of protein we want with the desired amino acid ratios. And so if we look at other ingredients here, such as poultry byproduct meal as one example, and we compare that to something like fish meal, we can see that these two ingredients have very similar amounts of crude protein. However, when you look at the fish meal, you can see that the fish meal is generally enriched in lysine and methionine in contrast to poultry byproduct meal, and therefore can be used to complement poultry byproduct meal and improve the ratio of lysine and methionine. A really great example of an ingredient where we tend to bring in methionine would be corn gluten meal, which also has a similar amount of crude protein as poultry byproduct meal and fish meal, however, is enriched in methionine, but is a poor source of lysine and arginine. So with these concepts, you can appreciate that what we really have to do is we have to combine ingredients to achieve the amount of protein we want in that formula and the ratio of amino acids. This becomes more and more difficult as you use fewer and fewer ingredients. My apologies, this came up the second time and not the first. Okay, and so when we combine these products and we make a pet food in whatever format that you'd like to imagine, the other component is that when we put these things together, they're not completely digestible. And so what we also want to do is account for the digestibility of those ingredients and the chemical reactions that occur when those ingredients are processed together. So cooking, canning, actually any kind of process that you can imagine. So you can hydrolyze proteins with enzymes, with acids, with bases. You can extract proteins, you can partially digest proteins. All of these things create a different digestibility of both an ingredient and a final product. Now, if we consider that we feed the animal 100% of any amino acid, and we account for the digestibility, which usually is somewhere in the range of 70 to 95%. So it varies very, very widely among different ingredients and different kinds of processes. What that does is it contributes to the amino acid pool in the body. Now, that amino acid pool is what is going to control how much body protein is synthesized. However, that can also be a repercussion of bioavailability, and I'm going to give you an example of bioavailability in a subsequent slide. And the other part is that amino acid pool can contribute to nitrogen excretion if those amino acids are oxidized, but those amino acids can also be used in metabolism. And we have recently shown, in particular with the amino acid tryptophan, that one such metabolite that tryptophan can affect is serotonin. Each amino acid has particular subsequent metabolic roles in the mammalian body. So now let's think about, we know what happens when you eat a food, you digest it, and you make that amino acid pool available. Well, the other component about that amino acid pool being able to support protein synthesis and what alters the availability to make other metabolites is the concept of the limiting amino acid theory. Now, this theory is predicated all around protein synthesis and supporting protein synthesis. And so let me help you imagine here that this barrel, what's going into the barrel, the water that you pour in, is the proteins that the body can synthesize, okay? Each stave or slat on this barrel is an essential or an indispensable amino acid. So what I have here in the yellow is methionine. And in this case, methionine, when we also account for digestibility and bioavailability, is only available to make protein at 50% of the required level. Well, what is going to happen here is it's going to truncate protein synthesis to 50%. And over a long period of time, what will happen is that animal will slowly mobilize both export proteins, such as albumin, which the liver makes, or structural protein synthesis, such as muscle and tissues. Those are structural. And so slowly the body will start to break those down in order to support whole body protein synthesis. However, to avoid that and to meet methionine requirements, we can supply DL-methionine as a supplemental amino acid in our pet food or in any mammal's food, and that would bring protein synthesis up to the next limiting amino acid. In this case, I've identified threonine as that limiting amino acid, but no worries, we also have threonine available and approved to add to pet food, so we could add that in to improve that ratio, and then we would then turn to the third limiting amino acid. So we have the capability of adding individual amino acids to optimize or improve protein quality. Now that I've described how protein content and amino acid composition contribute to protein metabolism, let's talk about what affects protein digestibility and amino acid availability. So although an ingredient can have sufficient total or crude protein, a good amino acid profile is needed to support whole body protein turnover, as I've demonstrated in the previous barrel analogy that I use. But what can also affect that bioavailability for amino acids to get into that central pool so they can support protein synthesis is digestibility, metabolic availability, and competitive inhibition. I'm going to describe a little bit digestibility and metabolic availability on the subsequent slides, but I'm going to describe competitive inhibition here. If you are if you're involved in the pet food industry or the animal agricultural industry, the best example of competitive inhibition would be calcium and phosphorus, and our focus on ensuring that ratio. In the case of amino acids share transporters at every tissue, and they also share a whole bunch of metabolic pathways. So a great example, and one around tryptophan, which we've recently published again, is tryptophan shares a transporter with what we call the large neutral amino acids. And those are the branched chain amino acids, leucine, isoleucine, and valine, and the aromatic amino acids, phenylalanine and tyrosine. And when those amino acids are really high, it literally prevents tryptophan from crossing into tissues so it can be synthesized into serotonin. This is most important when you think about tryptophan's ability to cross the blood-brain barrier and make serotonin. Why does this matter? Because serotonin controls circadian rhythm. Okay, and so here's just one example of a secondary metabolite that can be altered due to competitive inhibition. Now, in terms of digestibility of current ingredients in pet food, animal proteins are not necessarily superior to plant proteins. And it truly depends on what those plant proteins are, what the other compounds in those plant proteins are, and the level of processing that both the animal and the plant proteins have undergone. So it's not completely all animal proteins are superior to plant proteins. The other thing to remember is processing. So our example here that we generally use in pet food is the two big processing is either canning or extrusion. And I'm not denying that there are a myriad of other ways to process ingredients, but these are the ones that are most commonly used in the pet food industry. Cooking and drying both alter the bioavailability of amino acids in all ingredients and in final products. That means that if you're predicting your final amino acid composition and you're not accounting for potential process differences from day to day, that you might have a different bioavailability of amino acids in your final product. And so you should keep that in mind, that processing can affect digestibility and bioavailability. Then we need to think not just about how we're supporting protein synthesis, but the digestibility, the subsequent bioavailability also affects secondary outcomes. So the one that I keep coming back to here is serotonin. Now, the last part is that other animal nutrition sectors have values for ingredient digestibility, which are additive, and we do not have the same in pet. And we do not have a really good idea about how mixing together different types of ingredients may alter the digestibility and bioavailability of the final product. And we need to do more of that work to understand how to optimally process ingredients and then the combination of ingredients for the final product. Moving on to bioavailability, The best example of how bioavailability can be affected by cooking is what we call the Maillard reaction. Most commonly in the pet food industry, we think about either total and reactive lysine. That reactive lysine, when it's cooked, will bind with glucose to form a Maillard product. Now, the bugger about this Maillard product will be absorbed from the gastrointestinal tract. So you cannot capture it in measures of digestibility, whether that's ileal digestibility or total tract. For that, you have to move to measures of bioavailability, such as isotopic dilution techniques, to understand whether the lysine that's absorbed as a Maillard product is available for protein synthesis at the cell level. So last, we've defined that concept all around the characteristics of the ingredients and how we combine them to form the final product. But what ultimately defines how that final product's amino acid and protein composition can define protein quality is you have to compare that final product amino acid and crude protein content to the requirements of the species of interest. And you may be shocked at the lack of data that we have here. So in the case of methionine and cysteine, which I'm going to focus on today, there are 3 publications listed in the NRC and which were available prior to the last NRC's penning. And these all used immature beagles or pointers. Okay, and so we know that young animals have much higher protein and amino acid requirements than their adult counterparts. Second, they used a simple titration with a with a more chemically defined diet. What this means is that this is more digestible and bioavailable than any products that use intact ingredients that we use today. And what that might have done is that might have underestimated the requirement. However, what was commonly used to define the methionine and cysteine requirements was a weight gain, which is a really gross outcome in contrast to what we have available to us today. And because weight gain was used, this takes much longer to be affected than any measures that we have today, such as isotopic dilution techniques or metabolomics. And using weight gain may have landed us in an underestimation of amino acid requirements, and specifically methionine and cysteine. So you can see here that we likely don't have a really accurate definition of the methionine and cysteine requirement. The thing is, we struggle in the companion animal industry because we have to feed all these different breeds of dogs. If their phenotype is this different, it suggests that their genotype may be different as well. And then that begs the question, who do we target and who do we use to make good decisions to formulate our foods? When in fact, we've been using data from almost only this guy for the entire history of the pet food industry. So let me share a little bit of data with you to set up Sydney's last part. Just published earlier this year, and I will send this to the organizers so you can read this, this study if you'd like. We published the methionine requirements in small, medium, and large breed dogs, and these were greater than the current estimates of the requirement. But at the same time, the Waltham Center also published a methionine requirement paper, which resulted in a much lower requirement, and I'll explain that a little bit to you now as well. And so if you remember, I walked you through the methionine and cysteine about likely over-predicting, under-predicting, and under-predicting the requirement. But we also don't know anything about breed differences, interactions among nutrients, or how aspects such as dietary fiber may alter the sulfur amino acid requirements. And so we needed to tackle only one of those, and we tackled the breed differences first. So we measured the methionine requirements in miniature Dachshunds, Beagles, and Labrador Retrievers. And to acclimate you, here are the AFCO numbers, the FEDIAF numbers, and the NRC numbers. And I keep clicking as I did that, my apologies. Now, we could not define a requirement for the miniature Dachshunds, and what this suggests is that small breed dogs or miniature Dachshunds likely have a much lower methionine requirement than medium and large breed dogs. In Beagles, you'll see here, this would be the minimum requirement that would cover the mean of the population, whereas the upper confidence limit will cover the requirement of 95% of the population, accounting for inter-dog variability. The estimates of the minimum requirement and the confidence limit were similar among Beagles and Labrador Retrievers, so we pooled that to define a requirement for medium and large breed dogs, where while the mean requirement was similar to AFCO, the confidence limit was significantly than AFCO and similar but still greater than FEDIAF. Now, in contrast, Matt Harrison's group at Waltham also defined the methionine requirement, but only presented the requirement as a grams of methionine per megacal of metabolizable energy. And the first thing to note is that Europe uses the Atwater, not the modified Atwater, and NRC to predict energy density. So they actually come up with a higher energy density, which would suggest a lower requirement for methionine. So that's partly what accounted for these differences. Second of all, the feed intake between the two groups. Our group had significantly lower feed intake than the Waltham group. And it is important to remember here that while we're looking at amino acid requirements as a function of diet, amino acids are required on a grams per lean body mass basis on an individual animal. And so what likely happened here too is the estimate of Harrison's requirement is lower because those dogs were being fed more, in contrast the dogs on our study. So a little bit of nuances in terms of the differences in these two different studies, both very valid and both very important for the literature. So the take-home message here was that Beagles and Labrador Retrievers have greater sulfur amino acid requirements than miniature Dachshunds, and that the sulfur amino acid requirements may be greater than recommendations from AFCO and FEDIAF, but the total sulfur amino acid requirement still needs to be measured. Neither us nor the Waltham group has measured the ability of cysteine to spare the methionine requirement at this stage. Last, greater diet consumption may reduce the sulfur amino acid requirement. Now, these requirements, when we've done exactly the same approach in all amino acids, we used also the same dogs. So this work was conducted when I was still at P&G. And you can see here that in the case of phenylalanine, tryptophan, threonine, lysine, and methionine, our estimates are all greater than AFCO and NRC. And this is similar to what happened when state-of-the-art techniques were applied to human nutrition, and all the amino acid requirements came out higher than the original requirements based on the nitrogen balance technique. But measuring protein synthesis isn't enough. We need to think about behavior, immune status, body composition as well. And I'm going to give you one little snapshot about why that should matter when we think about methionine and cysteine. So that same study where we defined the methionine requirements, we used these diets with methionine ranging from 0.21 to 0.66. All the small, medium, and large breed dogs were fed exactly the same amount, and they had their blood taken at the end of each one of these studies. What's important to note here is, despite being fed exactly the same diet, Labrador Retrievers have significantly lower taurine than Beagles or Dachshunds. And this is additionally accounted for by having significantly greater methionine than Beagles and Dachshunds and suggests a fundamental metabolic difference between these dog breeds. So the take-home message here is that despite being fed the exact same diets as similar intakes, fed state plasma amino acid concentrations of methionine and taurine differed significantly among breeds and did not differ among dietary treatments. Now, it is important to acknowledge it was a short-term diet acclimation, and it might not have been sufficient to upregulate taurine synthesis in large breed dogs, as one example. Next, Labrador Retrievers have significantly greater methionine concentrations and lower taurine concentrations compared with Beagles and miniature Dachshunds. And as I stated, this suggests fundamental differences in sulfur amino acid metabolism among breeds. So with that, I'd like to turn it over to Sydney, who's going to take it from here. Sorry.
Great, thank you, Kate. As mentioned earlier, my name is Sydney. I recently completed my master's degree with Dr. Shoveler at the University of Guelph. However, before I joined the Shoveler Lab, a previous postdoc and several collaborators across multiple disciplines across North America wrote a review on the gaps in knowledge surrounding the potential link between the consumption of grain-free diets and canine dilated cardiomyopathy, or DCM. This review highlighted multiple areas of sulfur amino acid metabolism that require further investigation, including nutrient requirements, as Dr. Shoveler just spoke about, and about how the addition of micronutrients affect sulfur amino acid metabolism and taurine status. Next slide. So this was the topic of my master's research that I will be discussing today, specifically looking at the addition of dietary supplementation of taurine, methionine, or methyl donors and receivers, and how those affect sulfur amino acid metabolism and taurine status in the beagle. So before I go any further, I just wanted to give a brief overview of sulfur amino acid metabolism and some of the precursors to taurine synthesis as well. So up at the top in green, we have methionine, which is consumed in the diet. Methionine can be converted to S-adenosyl methionine, or SAM, which is known as the universal methyl donor. SAM can donate a methyl group to create a variety of different products, but three of which will be the main focus of today will be creatine, phosphatidylcholine, and carnitine. After donation of a methyl group, S-adenosyl methionine is known as S-adenosyl homocysteine, which can then be converted to homocysteine, at which point homocysteine has one of two fates. It can either be remethylated back to methionine, shown in red there, via methyl donors choline via betaine, or folate. On the other hand, homocysteine can be irreversibly converted down to cysteine, shown in orange there. And cysteine has a variety of different fates, one of which being glutathione, shown in purple, which is known to have antioxidant capacity. And then down at the bottom there, one of the fates of cysteine is taurine. And as we know, taurine plays a major role in contractibility of cardiac tissue, as well as maintaining osmotic conditions in the heart. It's not exactly known how it does that, but it is known that it plays a major role in the heart. And taurine deficiency linked to dilated cardiomyopathy has been shown to be more prevalent in large breed dogs, and that's possibly due to a lower taurine biosynthesis rate in these large breeds. So as shown on the previous slide, there are many other upstream metabolites of taurine that are often overlooked in companion animal nutrition, but have been studied in other species quite extensively, such as in piglets. So when fed a methionine restricted diet, supplementation of all three methyl donors, folate, choline, and betaine, provided 8% more methionine in piglets. Another upstream metabolite that I mentioned, homocysteine, has been correlated with heart disease in humans when at elevated concentrations, known as hyperhomocysteinemia, and fairly new research has suggested this could be possible in dogs as well. So the objective of our study was to investigate what happens to these upstream and downstream metabolites and amino acid concentrations in dogs after being fed a grain-free diet supplemented with either methionine, taurine, or methyl donors and receivers, creatine, carnitine, and choline. And in order to do this, we used 8 beagles that were pair-housed, and the study was a 4x4 Latin square design, meaning each pair of dogs received each treatment. Again, we had 3 treatments and a control, and the control diet was a commercially available grain-free diet. And then the 3 treatments being either methionine, taurine, or creatine, carnitine, and choline. So each period lasted 7 days. And then on day 7, the dogs were catheterized and a series of post-meal blood samples were collected. And we measured plasma amino acids as well as glutathione at 0, 15, 30, 60, 90, 120, 180, 240, 300, and 360 minutes following the meal. So before I move on with the results, I'll just orient everyone here. So I'll be presenting a series of these figures with time along the x-axis in minutes, and then plasma amino acid concentration along the y-axis. So starting first with results from plasma methionine, we found a significant time by treatment interaction effect, where dogs supplemented with methionine had significantly higher plasma methionine concentrations from 30 to 360 minutes following the meal compared to the other 3 treatment groups. And this was expected since the methionine was provided in addition to the diet and was therefore readily available to each dog. Similarly, for plasma homocysteine, we found a similar time by treatment interaction effect, where dogs supplemented with methionine had significantly higher plasma homocysteine concentrations from 60 to 360 minutes following the meal compared to the other 3 treatment groups. And results for plasma taurine, although we did not find a significant interaction effect, we did find both a significant time effect and treatment effect. So on the left is the time effect there, and plasma taurine concentrations were significantly lower at 15 minutes, but significantly higher at both 60 and 90 minutes post-meal compared to fasted concentrations. And on the right, we have the treatment effect, where dogs supplemented with taurine had significantly higher plasma taurine concentrations compared to dogs on control. But interestingly, dogs supplemented with either methionine or creatine, carnitine, and choline were actually not different from the dogs supplemented with taurine. So in summary, methionine is equally as effective as a combination of carnitine, choline, and creatine at supporting taurine status, but only taurine improved taurine concentrations over control. And it's likely that the methyl compounds can spare the use of methionine for methylation and allow methionine to enter the transsulfuration pathway while keeping homocysteine concentrations the same. And this is important as we continue to do more research that may suggest a possible link between elevated homocysteine and heart disease in dogs. So now that we've covered the micronutrients, I just wanted to spend the last couple of minutes here briefly discussing the results from our global survey. And that is published in PLOS, and I believe it is a handout as well. And that was our Grains on the Brain, a survey of dog owner purchasing habits related to grain-free dry dog foods. So I don't have too much time to go into all the details, but there is a lot of results there. So if you're interested, please take a look. And this survey was, as well as the previous study, was funded by the R.C. Hagen Company, which is a Canadian pet food company. And the objective of our survey was to identify the factors that are predictive of a dog owner's choice of grain-free dry dog food across Canada, the US, France, Germany, and the United Kingdom. So I'll just briefly highlight our major findings from multinomial linear regression. And we found that people who look for dog food with no grain are more likely to be female, to believe their dog has an allergy, to follow 5 or more of their own dietary regimens, to not include grains in their own diet, to look for more protein options in pet food, as well as no fillers and no byproducts, to give more other foods to their dogs, such as treats and table scraps on a daily basis, to rank ingredients as more important, to static feed their dog or not rotate their dog's kibble. And finally, to get their information about pet food from pet store staff and online, and purchase their pet food from pet specialty stores, veterinary clinics, and online. So this survey taught us about the attributes that make the grain-free consumer unique compared to the grain-inclusive consumer. And it's important to understand not only the metabolic changes in dogs consuming a grain-free diet, but how and why consumers are choosing to feed a grain-free diet, and if those decisions that the pet owner is making at home may put their animal at risk of developing any secondary metabolic disorders, such as DCM. So finally, I will pass things back to Kate just to wrap up.
Great, thank you, Sydney, and thank you, Balcom, for inviting us to share our research and our insights on pet food. My lab and myself and all of my collaborators are always very willing to work with the pet food industry. While I have conflicts of interest, I believe that we have a common interest, and that is to make the world a better place by supporting the health and well-being of pets, by supplying a wide range of foods that meet both the consumer needs and the pet needs. So with that, we'd love to take any questions that you might have. Thank you.
Well, thank you, Dr. Shoveler, and thank you, Sydney. But before we get started answering questions, we'd like to share a brief video, and then we'll be right back to answer the questions submitted during today's presentation.
At Balchem Animal Nutrition and Health, we strive every day to deliver results you can see in your animal's productivity and your bottom line. From a smooth transition into the milking string for your fresh cows, to a happy welcome home from your furry friend. From a strong start in your poultry flock, to consistent weight gains for your finishing hogs. We expect to earn your business and your trust with our people, our products, and our science. Our people have an intense passion for your animals and your success. You can count on us for honest, candid advice and practical solutions for your toughest challenges. As the global leader in choline production, chelation, and encapsulation technology, we take our obligation to you and to the environment seriously. Our products are backed by the most extensive and thorough research portfolio, while our business is committed to advancing environmental sustainability and animal welfare. In the end, it all comes down to results. Balchem delivers real results you can count on, results that exceed your expectations, and results that bring true value to your bottom line. Leading the charge to meet the nutritional needs of ruminants, monogastrics, and companion animals, Balchem offers a growing portfolio of nutritional products and a dedication to innovation and industry sustainability. Balchem is here to solve today and shape tomorrow.
As a reminder, you can still submit questions to the questions pane in your attendee control panel. Kate, your first question is going to be, let's see here. Eduardo is asking, is there a mechanistic basis to the link between DCM and homocysteine?
That's a great question. So homocysteine is a pro-oxidant, and that would be one connection. The other is likely that it's a marker of poor remethylation of homocysteine to methionine or transsulfuration, which largely occurs in the liver and the kidney. And so there may be a additional role there that if that links into lipid metabolism. So homocysteine also tends to go up when cholesterol and low density lipoproteins go up as well, which are also, as you know, indicators of cardiovascular disease. But whether that is direct signal has not been elucidated to my knowledge.
All right, thank you for that. The next question is, do you think digestibility values are needed for dogs and cats foods? And if so, is a total tract okay to look at amino acids?
Great question. So total tract protein and amino acid digestibility does not represent that which is available to the animal. So if you truly want to understand amino acid digestibility, you either have to go into the swine ileal digestibility literature, or you have to go into the cecumized rooster digestibility research, and those would be most representative of amino acid bioavailability. Maybe as a teaser, my group is currently trying to put all of that data together in a meta-analysis to provide to the pet food industry.
All right, thank you. Matthew is looking for a recommendation in TSAA for methionine to cysteine ratio, as well as other ratios, lysine to threonine, lysine to tryptophan.
Okay, great. So he clearly is going into we definitely look at these when we go into the swine literature. Now you can do those by putting the combination of the amino acid requirements together. And so I can tell you, as an example, we looked at the ratio of tryptophan to large neutral amino acids. And off the top of my head, I'm not going to be able to give you those numbers, but please give me an email at Guelph, and I'll quickly pull those up and shoot them to you. But right, you can actually do a ratio across at least 4 or 5 amino acids, and likely methionine is your better ratio amino acid in this case. It tends to be the first living amino acid across all kinds of ingredients for dogs. So I might use that as a ratio for my other amino acids, but whatever ratio you use, it would be an improvement. So off the top of my head, I don't have the numbers. And your other question about methionine to cysteine, when you look across the literature and across species, it is generally about a 50-50 ratio on a weight basis. It's different on a molecular basis, or on a molar basis, sorry, not a molecular.
All right, thank you. Jackie is saying wonderful presentation. In Sydney's study, did you attempt to define marketing terms like fillers, no byproducts, etc., or did you rely on pet owners being familiar with these marketing terms?
I'll take that one.
Yeah.
No, we intentionally did not define any of the terms that we used because we wanted to know what consumers are currently doing based on the knowledge that they have, and not us providing any kinds of definitions or anything like that would influence their responses. So we actually didn't even use the term grain-free at all during the survey. We only used the term no grain for that exact same reason, to prevent adding in any biases towards their responses.
All right, thank you, Sydney. Caitlin is asking, do you think more research needs to be done on how processing affects the digestibility and bioavailability of nutrients, specifically amino acids? Is there current publications on how much loss there is currently?
So yes, the answer, well, it's unfair to ask an academic whether they think that more research is needed, because it's an endless path of unanswered questions and challenges to assumptions. So the answer to, do we need more research, is absolutely. And I think that's fundamentally, it's likely that both digestive capacity and metabolic capacity is different among different breeds of dogs. And then if we throw the cat in there, all bets are off. The vast amount of processing work that's being done with regard to digestibility is being done at Kansas and Illinois, both highly reputable groups that I highly support. And so that is emerging. I would also encourage others to go and look across the ag industry for these data. So in particular, I have a fairly large project, which is my Agriculture and Agri-Food Canada grant, in combination with Sask Pulse Growers and the University of Saskatchewan, where I pulled in the brilliant mind of Rex Newkirk to help us with processing and understanding how different types of processes affect nutrient availability in novel pulse ingredients. But that is the kind of work that's also being done elsewhere, and is needed, especially with some of the criticisms that we've gotten with not understanding that well enough.
Thank you. Angela has a couple questions. The first one is, when you said that the different breeds of dogs were fed the same amount, is that the same total amount of food or adjusted for body weight?
Yeah, adjusted for body weight. We fed them a similar amount on a per kilogram basis. Yeah.
All right. And the next one, she's asking, legume content has been discussed in the industry regarding DCM. Just curious if the grain-free diets used was high in legumes.
I'm assuming that's for the Beagle study. So I'm not sure the exact, I guess, percent of legumes in the diet, as it is a commercially available diet. However, both peas and lentils were in the first 5 ingredients of the diet.
All right. The next one comes from Carrie. What could amino acid imbalances create?
Yeah, great question. So probably one of the most profound issues that it'll create, the imbalances, is it'll create a deficiency of another amino acid, despite it looking like your intake is over the requirement. So if you had a really disparate ratio, but everything was over the requirement, you actually may be creating a deficiency of your lower amino acid by default at the tissue level. And that could also go the other way, where you're creating a potential amino acid toxicity as well. And amino acid toxicities are not very well understood in any mammal. And the other part is amino acid imbalances are also not well understood, and that's because there are very few industries, and ours is the one where animals don't have a diversity of what they're eating. So even in animal agriculture, there's a diversity in the different growth, in the different phases of diets that animals will receive during growth. And even there, what we do see, however, is a balancing of the amino acid ratio. So we look at swine and poultry, which would be comparable to dogs, and amino acid balance is a primary formulation target for those industries. And so not balancing your amino acids and having the wrong ratio is likely then to create either a toxicity or a deficiency of another.
All right, great. I think you've addressed this question, but I'm going to ask it again. Eduardo would like to know, are these grain-free diets based on the use of legumes? And then what about meat-based diets? Have those been looked at?
You answer first one, Sydney. I'll answer second.
Yeah, so grain-free diets, yes, they have no grains. They're replaced with legumes. So peas, lentils, chickpeas, beans.
I think the other important thing to layer on to that is grain-free diets, because legumes are used in contrast to grains, is also to remember that it tends to result in a much higher protein content as well. So that would be a marked difference between grain-free and traditional diets. And sorry, I hate I actually kind of dislike a lot of the nomenclature we have, because it's a lot more widespread differences than that, and yet we're bucketing diets that are completely different in the same bucket. And I think we need a little bit more accuracy when we're talking about diets. The second part of the question was, is there research on meat-based diets? So we're seeing a lot of, well, it depends on exactly what you mean. But I think traditionally, I would say, is that we have a snot ton of data on feeding chickens, or feeding chicken to dogs, but we have less information on other animal products, and it, you know, and the relative amino acid digestibility of those as well. Now, what we are seeing, especially from Kelly Swanson's lab, he's doing a lot of work with kind of those fresh, lightly cooked, and looking at the digestibility of different meats in those. And so that might be kind of a good place to start, with a caveat that we know that those are lightly cooked. So remember my warnings about processing and cooking and the variability that that may produce in terms of the digestibility of the amino acids.
All right, good answer. The next question comes from Xavier, and it's for Sydney. What was the total amount of methionine of commercial food and the quantity supplemented in your study?
Yes, I have the numbers right here. So we added an additional 2.6 gram per kilogram dry matter. So that's what I added to the dog's diet on top of the diet each day. And then the total, including the diet, was 7.2 gram per kilogram dry matter. So that was including supplementation and the base diet.
All right, thank you, Sydney. And the next question then comes from Sarah. Amino acid values are not reported for most store-bought dog foods. How do you know they're receiving adequate levels?
Fantastic question. I agree. You don't, as a consumer, and you put your trust in the companies that are reporting those. And so in the United States, you do have to submit your typical analysis, which includes amino acids as part of the AFCO profile. Here in Canada, you don't have to register your products, but what we've been doing, and I'm not sure, nobody is phoned to yell at me yet, but myself and my colleague at Joni Verbrugge's lab have been going through commercial products and analyzing them for amino acid content now, and looking to see whether that content exceeds recommendations. And in some of those that we did a made in Canada a couple years ago, and I believe it was threonine that popped up as coming out below AFCO in a number of diets. And more recently, in a, I think it was an abstract being presented and a manuscript being prepared, my colleague, Dr. Verbrugge's lab, has demonstrated that in plant-based diets, and not surprisingly, that methionine keeps coming out under the recommendations. So you need to rely on academics to go out and literally analyze those products, or you are dependent on your state officials to get them, and to demonstrate that they were adequate. What you can do, and what veterinarians are told to do, is that they are told to phone the company and ask for the typical nutrient analysis of the product that they're considering. And I believe that that wording was recently changed in the WSAVA recommendations, or I think they actually changed it as well to guidelines for veterinarians for selection of pet food.
And we've just got to the top of the hour, so we've got time for one more question. Should nutritionists develop formulas on an amino acid requirement basis rather than current formulation strategies? Increase levels of protein, meat first, etc.
I think whoever's asking me that question probably knows what my opinion is. I think that the consumers see ingredients, and this is really about knowledge translation. The consumer doesn't want to see amino acids. They trust that we're putting the ingredients together that they want to eat to provide them the nutrition that they need. And so here I would remind everyone, remember that animals don't require ingredients, they require nutrients. We put ingredients together to meet their nutrient requirements. When we look at human nutrition, if you look at, I believe that your human guidelines in the States use the plate, right? We use the pyramid, use the plate. I think that's right. Nobody's answering my question, but I think that's right, or maybe they're changing. But when you put the plate together, and if we went and calculated that by following recommendations for diet diversity, it's hitting macro and micronutrient requirements, and it's the nutrient requirements that underpin what ingredients are being suggested to humans. And we need to think about that as well, because we're feeding static diets. Humans change what they eat every day, so we're less worried about them meeting nutrient requirements. We're feeding dogs the same thing, so absolutely, you should have target nutrient requirements for your entire line of products, despite what ingredients that you're using.
Excellent. Thank you for the webinar today, ladies. Thank you both, and thank you everyone for attending today's webinar. If you have additional questions, please submit them to anh.marketing@balchem.com, and we'll forward them along with the unanswered questions from today's session. Our next webinar will be delayed by 1 week due to the 4th of July holiday. On July 13th, we will hear from Dr. Charles Hofaker from the Southern Poultry Research Group. He will review the newest research around nutraceuticals in poultry. For our ruminant audience, on July 20th, Dr. Kevin Harvatine from Penn State University will take us through the impact of daily rhythms on milk and component yield. Visit balchem.anh.com/realscience to learn more about the next companion animal webinar and to access past event materials. A recording of today's webinar will be available within 48 hours, so you can revisit this content and pass it along to your colleagues. Balchem's podcast series continues to offer a deeper dive into our webinar topics. We now have 15 episodes out on YouTube, your favorite podcast platform, and also at balchem.anh.com/podcast. Our latest podcast features Dr. Greg Aldrich and several students from the pet food program at Kansas State. A new episode will be posted tonight, so be sure to subscribe to the Real Science Exchange. On behalf of Balchem and Dr. Shovler and Sydney Banton, thank you for joining us today.
Thank you.